Well, thanks very much for coming. In the next 45 minutes or so, I was going to share with you some of background information on arsenicals and the kidney, some work that we've begun with Dr. Athar's group in dermatology, and some perspectives on where this area may be going. So again, wanted to acknowledge this UAB Research Center of Excellence for Arsenicals that was just funded recently by the NIH, and I'll talk a little bit about this NIH CounterACT program as well. So these are my disclosures for the CME for today. So here's my outline. I'll talk a little bit about a historical perspective. We'll talk a little bit about chemistry of arsenicals, arsenicals as chemical weapons, and I'll preface that with some broad introduction on chemical weapons, historically where they've been and where they're going. Arsenicals and the kidney highlight more acute and chronic kidney disease, and then a little bit about this NIH CounterACT program. So I'm sure you all know who this is. Okay, so you got the first question right, but I will come back later. You know, does anybody know, at least only the trainees maybe, how he died some 200 years ago? Not anybody from the U54 group either. So what did you say, food poisoning? Arsenic poisoning? Arsenic poisoning? Okay, we'll address that when we get back if that was correct or not. So arsenicals, you know, it's in the periodic table, that's why the symbol AS33. It was commonly used in a lot of alloys to make them more strong. For example, lead, bronze, all of them had arsenic in them. It's a major component of the Earth's crust, so there's a ton of arsenic in the Earth's crust, and that's led to some problems, which I'll share with you in terms of contamination of groundwater. It's a semiconductor, so it's used in a lot of the semiconductor industry, in the Silicon Valley and so on. It's used commonly as pesticides, as herbicides, as insecticides. And one of the chemicals, arsenic trioxide, is actually FDA approved for chemotherapy for acute promyelocytic leukemia. And it's used in combination with known drugs for better remission rates in these patients. In old ages, they used to use arsenical derivatives to improve the skin complexion. So either they would eat arsenicals or they would rub it on the face or the skin to make sure that the skin, you know, became more fair. And it was known for a long time as the poison of kings. And the reason for that is it was a very slow poison, the way it was ingested or put in drinking water. So people would die very slowly. So it was very potent and also discreet. So nobody would know if you just slipped it into your wine or, you know, your food or drinks, you know, over a long period of time. Nobody would know. You know, it didn't have any specific way of recognizing it till, you know, about 200 years ago. They developed tests, chemical tests that kings would have their courts perform to make sure the food was not adulterated with arsenic. That was called the Marsh test. And it's a simple chemical reaction that will tell you if any food or drink would have arsenical, and the king, you know, would not, you know, ingest that. So really a lot in the history in terms of arsenicals being used going into ancient times. So here are some of the structures you can see, some of the derivatives mainly. You know, this is arsenic trioxide, the drug that's used for leukemia. Arsenic trichloride, arsine, which is a highly poisonous gas, and I'll share with you a case report on a person that was exposed to arsine gas. And some of the other chemical weapons that I'll talk about is lewisite, diethylchloroarsine, diphenylcyanoarsine, diphenylchloroarsine, and this molecule, phenyl arsine oxide. This is actually a surrogate molecule for these arsenicals. So if you were to do research using any of these chemicals today, you cannot do this in your lab, even if it's BSL, you know, 2 or 3. You have to go to Kansas City, where there is a governmental agency approved called MRI Global, and they are the only agency in the country that can do any research that is relevant to these arsenicals listed on the slide here. However, this POW, which is a surrogate for these arsenicals, is not as toxic or a bioterrorism listed chemical that one can use it. So we use it in the lab when we cannot, we want to just reciprocate or duplicate our studies using these highly toxic arsenicals that are essentially bioterrorism compounds. And Lewisite was developed in the early 1900s before World War I as a chemical, put in a lot of bombs and so on to cause extensive skin blistering and inflammation and death of a lot of people. And the British developed British anti-Lewisite, essentially a molecule to prevent toxicity of Lewisite. And I'll show you some of the slides along those lines. So how well can one get exposed to arsenics? So you can get exposed to arsenic with the drinking water. You can inhale arsenicals if you're working in these industrial areas which make arsenic for different purposes. You can ingest it either in food or you can also have cutaneous exposure through your skin. And as we know, the skin is the largest organ in the body, so therefore based on the surface area, that's a tremendous source of exposure to arsenicals. So this was a big story several years ago, and unfortunately is still a big problem. So you all know where Bangladesh is. It was earlier called East Pakistan. Is one of the countries just situated on the east part of India. Population of about 125 million. And it's thought that over half of these people in this country are exposed to drinking water that is hugely contaminated with arsenicals. And the reason this occurred was because of the water shortage, and the water from the rivers and so on that were higher level were not very pure. So the government decided to bore tube wells. And this is an example of a tube well. So it's a small metallic tube that goes about 80 to 150 feet deep into the water table, and then you can use this as a pump and essentially get fresh water. In fact, growing up in India, when we visited North India, my family and so on, every home would have these tube wells. And I remember, using the pump, you can get just fresh water, coming out. It's nice and cold because it's, from the bottom of the earth, 150, 200 feet down. And Bangladesh itself had 4.9 million tube wells. So almost every home had a tube well installed. And because of the depth where these tube wells were using the water, to get pumped out, that was heavily contaminated with arsenic in the Earth's crust. And with number of years, with all the bacteria and other organic decay, that arsenic, was released and contaminated the water. So the levels of arsenic in the water was at least 5-fold higher that the people were drinking in these populations. So over several years, and this was detected in the late 80s, and they have sampled a lot of these wells through work of the WHO and the local government to try and make sure that they shut these wells down. So this is actually a well that's been shut down, so you cannot really pump water out of it. But there are several that are still using these tube wells, as a source of water. And it causes a lot of havoc. In fact, this was from a bulletin, WHO bulletin in 2000, but as of 2018, that several wells were still in existence that were contaminated with arsenic, and levels of over 500 microgram per ml of arsenic in those wells. And some of the diseases that would occur was like this, a skin disease, keratosis, and this occurs from chronic exposure to arsenicals, and it predisposes to skin cancer. Not only skin diseases, but these patients have bladder cancer, renal cancer, because a large way of how this arsenicals are excreted are through the kidney, and therefore the kidney, the urinary system gets exposed to this carcinogen, and you can get these cancers in different organ systems. The way arsenic gets absorbed is through intestinal transporters, mainly aquaporin 10, GLUT5, and organic anion transporting polypeptides. And these same transporters are actually present in the kidney as well. So nobody knows how much of it is apically taken up. But there's a lot of work done in terms of basolateral uptake. Various transporters listed on the slide, GLUT1, GLUT5, aquaporin 3, this OAT. Apically, there's also export mechanisms, both apical as well as basolateral. And these are involved with a lot of heavy metals and drugs in the kidney. For example, export of mercury, cadmium, cisplatin, and so on. They all occur through the same system. So there's not very specific for arsenicals, but it's broad transporters that are used for heavy metals within the kidney. So the half-life is very short, in humans less than an hour. There are major detoxification systems within the body. For example, these metallothioneins, these are cysteine-rich compounds that bind to these heavy metals, including cadmium, mercury, and so on. And these molecules, arsenics in particular, can uncouple oxidative phosphorylation, particularly in the proximal tubules that are highly metabolically active, and you can get dysfunction of the proximal tubule. For example, you'll have failure to absorb phosphate, glucose, amino acids, and those will leak in the urine, giving rise to a Fanconi-like syndrome. It can also cause direct podocyte injury, and you can get proteinuria or albuminuria. There have been a lot of instances of accidental exposure to some of the arsenicals, in particular arsenic trioxide, and as well as arsine as a gas, that can cause hemolysis and acute kidney injury in industrial workers. And some areas where the arsenicals have been contaminated in the water with very high levels, like what I showed you in Bangladesh, there's increased mortality, diabetes, chronic kidney disease, as well as cerebrovascular disease. It's been not clear. It's mainly an association. You cannot really say cause and effect, that this was due to arsenical exposure. But they've looked epidemiologically that if you have arsenic and cadmium exposure from drinking water and so on, or from soil, then the chances of kidney toxicity in terms of chronic kidney disease is much higher in those populations. Acutely, when you get exposed to arsenicals, you can get hypotension, shock, direct effects of arsenic on the proximal tubules, hemoglobinuria, podocyte damage, proteinuria. You can get a tubulointerstitial nephritis as well as a Fanconi syndrome that I spoke about. So here's a case report, and this was in the materials that Lisa had sent out. A 51-year-old man develops nausea, vomiting, diarrhea, abdominal pain, and dark urine 2 hours after inhaling a noxious powder at an industrial galvanizing plant. 2 days later, he had low urine output, striking orange pigmentation of his skin, sclera, as well as pedal edema. His hemoglobin was 5.8, so he was severely anemic. His platelet counts were normal. His creatinine had gone up to 6.8. His bilirubin was 1.9 with the direct bilirubin of 1.1. His retic count by day 7 had increased to 3.5%. His liver enzymes were elevated. His LDH was elevated. He had worsening kidney function. He developed proteinuria. His urine sediment was fairly innocuous. His ultrasound showed enlarged kidneys. So they treated him with steroids, blood transfusion, and hemodialysis, and then a kidney biopsy was done at 10 days. So here's his kidney biopsy. You can see his glomeruli showing mesangial proliferation and otherwise really normal. Panel B, you can see a lot of casts and acute tubular necrosis. And then this is staining for hemoglobin. So a lot of these casts actually stain for hemoglobin, and these dots here represent hemosiderin deposits, mainly in the proximal tubule, and when they stained this for iron, all these tubular cells were positive for iron as well. So really acute kidney injury in this patient from exposure to a heavy metal. So this case turned out to be arsine gas as one of the industrial pollutants that was produced in this galvanizing industry and caused acute toxicity, which has highly associated with the fatality of 25–30%. So 1 in 4 patients will die if they get exposed to this compound. This patient fortunately survived, regained complete kidney function back to normal at least a year after follow-up. So here's a cartoon just showing the potential way how arsenicals can cause acute kidney injury. So you get exposed to arsenicals, it depletes glutathione and related enzymes, it increases your apoptotic markers like caspase-3, increases cytokines IL-6, IL-8, increases DNA damage markers like p53, cause apoptosis. You can get oxidative stress, free radical generation, decrease repair of DNA, and cause proximal tubular injury. So I'll show you some work that we've done in collaboration with Dr. Athar in a mouse model showing that a lot of these pathways are clearly involved in pathogenesis of acute kidney injury with arsenical exposure, at least in animal models. We'll talk a little bit about arsenicals as chemical weapons. I told you about the development of lewisite in the early 1900s during World War I. There's been a lot of history to these chemical weapons. So in 1675, so a long time ago, there was the Strasbourg Agreement where they would use bullets or arrows that had poison on their tips. So the Strasbourg Agreement was developed to prevent that from occurring, that you could not use chemical weapons in bullets or in arrows to use. There was a Brussels Convention like 200 years later, again for using poison weapons during wars. The First World War, there were 1.3 million casualties from chemical weapons alone, and 90,000 of them were actually died due to chemical weapons. A lot of this at that time was lewisite, other gases like sulfur mustard, and so on. 1925, there was a Geneva Protocol to have a worldwide ban on chemical weapons. In 1930, there was a big exposure of chemical weapons in China with several people dying. 1972, there was a Biological Weapons Convention. 170 countries participated in it, 170 parties, but there were only 10 people who signed that as of 2014. So significant issues there. We know the Iran-Iraq War, the first Iran-Iraq War in 1980, extensive use of chemical weapons. 1993, there was another chemical weapons convention. And in 1997, at The Hague, they developed this program for chemical weapons prevention. And they started working, it did a really good job, 182 members. They actually destroyed 25,000 metric tons of chemical weapons. 3,000 inspections were performed. During their 10th anniversary was in 2007. And actually, 1998, 2013, they received the Nobel Peace Prize. And we know since 2013, the Syrian civil war, there have been a lot of chemical attacks. And this is just shown here as an example. You've seen more gruesome pictures on CNN and other news channels. So very unfortunate. And this just shows you the various attacks in Syria, different areas by either the Syrian government or ISIS or unconfirmed sources. You can see the red circles is Syrian government, going from 2014 in various areas of Syria, Damascus, Aleppo, Hama, and so on. And the size of the circle tells you how many were injured in each attack. So the biggest size of the circle means 1,000 people plus died in that chemical attack. And you can see huge number. So this was 85 attacks as of 2018, but 2019 there have been 200 chemical attacks just in Syria alone. So a huge problem, despite the world trying to limit use of chemical weapons. This is still a major ongoing issue. And this is a picture of this Organization for Prohibition of Chemical Weapons at The Hague that was recognized with the Nobel Peace Prize in 2013. So arsenicals have been used as chemical weapons, as I told you. They're highly toxic. They were used World War I and II. And this is the prototype molecule, lewisite, 2-chlorovinyl dichloroarsine, and the chemical structure is shown here. They're very potent vesicants. So if you get exposed to lewisite, you'll get extensive blistering, you'll get pain in your skin, inflammation, as well as if your eyes get exposed to this, you get extensive pain, blistering, conjunctivitis, and so on. And just cutaneous exposure to this can cause systemic injury. And I'll show you some of that in our animal models, and can affect the kidney, can affect the lung, liver, brain, and other organ systems. So really a systemic poisoning with cutaneous exposure to arsenicals. So during World War I, they would circulate, give these masks out to people saying, hey, wear this mask if you feel something like geranium, and this was a flower. And those of you who've smelled geranium, the smell of arsenic or lewisite is very similar to that. It's like a garlicky, like odor. And just as a protectant, that if you smell any gas during a bomb attack and so on, make sure you have this mask on so you can limit at least from inhalation or exposure to your eyes. It says, nasal irritant, skin burns, gas mask, protective clothing, and maybe a dark green oily liquid depending on how it is used. So a lot of preventive measures that people had tried to prevent citizens from getting exposed to these chemicals. Other examples, environmental exposure. So this other chemical, diphenyl arsenic acid, from chemical weapons decay in Japan. So there was a naval base that was producing these chemicals long time ago, but they had stored them and they had decayed and had actually gone into the water table. And again, contaminated water table led to people drinking that water and getting neurological symptoms and chronically mental retardation. Accidental exposure, beer bottles containing mustard and lewisite were unearthed at a road construction site in Japan. And laborers who were working in that construction site on the roads ended up getting severely exposed to lewisite. Other areas of Chinese victims that were exposed to sulfur mustard and lewisite from abandoned World War II vehicles, and you can see some of these old abandoned weapons that were weaponized with these chemicals and just left, and people not knowing they were buried somewhere, and then they were playing with it and they see but actually had arsenicals within them. And these are just some of the skin lesions that you can get from exposure to lewisite. So the work from Athar's lab, he's really pioneered this work at UAB, not just at UAB but in the entire world. Very pioneering work on arsenicals and how they cause skin injury. And we started, when he started exposing his animals to arsenicals, he noticed that they would develop multiple organ involvement. And we met maybe 3 years ago, Athar, in 2015 or so, 4 years ago, and said, can we collaborate and work together on this project? And we submitted an R21 grant at that time. Didn't get funded, but we had very good feedback, and that really led us to collaborate and have others from Division of Pulmonary Critical Care, folks from Southern Research, as well as the School of Public Health and Health Professions in developing this programmatic initiative that I'll talk about. So in this work, Athar's group really showed that using lewisite, using phenyl arsine oxide, I'll abbreviate it as PAO or PAO, that you could actually mimic what would happen with lewisite exposure in animal models. And he could dissect really the mechanism of how skin injury would occur in this setting. So these are mice where he did initial studies looking at multiple strains of mice. We know that mice have different susceptibilities to injury. For example, acute kidney injury. If you do ischemia reperfusion in a B6 or a BALB-C or an FVB, the susceptibility is very different. So to first elucidate what is the strain susceptibility to exposure to PAO, he did these studies using B6 mice, FVB mice, these sketch 1 mice, and these patch 1, sketch 1 mice. So these patch 1 mice are mice that have a mutation in the patch 1 gene, and they're bred with these SKH1, which are hairless mice, and it has impaired signaling in the hedgehog pathway. And these mice shown here before exposure, and then you expose it to PAO, and you can see within 8 hours the skin is significantly inflamed and thickened, and you can quantify that using this Drey score, which is used for quantifying inflammation in the skin as well as the eye. You can see these FVB mice as well as B6 mice are relatively resistant to injury, but the patch 1, sketch 1 mice are significantly susceptible to this injury. And you can see the change in skin fold thickness as well is significantly worse in these patch 1, sketch 1 mice. And that was the rationale for us to use these patch 1, sketch 1 mice in our studies. And you can see here in the murine model significant inflammation with lewisite as well as these other two arsenicals, DPCA as well as DPCYA. You can see histology of the skin with this blistering, significant inflammation compared to the control, increased the Drey score, the skin fold thickness is significantly worse in these animals that got exposed to the arsenicals. And this was nicely translated into a large animal model. using pigs. You can see here again lewisite as well as these other two arsenicals. Compared to control, there is significantly more inflammation. And if you do histology on the skin, again very significant blistering and inflammation, these microvesicant formation with fluid under the skin, epidermis, in these animals that were exposed to lewisite. So not only in mice but also in large animal models you can see significant inflammation in this setting. They also looked at the mechanism. So significant increase in inflammation. You can see markers like macrophages were significantly increased compared to controls. Myeloperoxidase, which is another marker for inflammation, significantly increased number of inflammatory cells with myeloperoxidase positive cells. And the same thing translated nicely in the porcine model of animals that were exposed to lewisite as well as these related to arsenicals. So they've looked at more detailed molecular analyses of the skin, and shown here is just a snippet of work looking at a volcano plot showing differentially expressed genes with lewisite. You can see so red reflects upregulated genes. There were 605 genes, and the blue reflects downregulated genes in this volcano plot. And the top hallmark gene sets are shown on the bottom panel, where you have TNF signaling, inflammatory response, as well as the complement pathway and the number of genes involved. So significant number of genes involved in these individual pathways. So the question we raised when we saw all this data is, are these arsenicals causing kidney injury? So we first started looking at acute kidney injury in this setting. So the question was, does cutaneous exposure to arsenicals cause acute kidney injury in mice? So what we did was, based on the data from Athar's lab, we used these patch 1, sketch 1 mice for the exposure to lewisite and related chemicals. These studies were done at MRI Global in Kansas. And we also used B6 mice to expose mice to phenyl arsine oxide, or POW. And we used lewisite at a dose of 1.5 milligram per kilo. And this is about 1/10 the LD50, which results in death of these animals. And these were done at MRI Global. And the surrogate, we did those studies here, and we used 150 microgram per mouse. We did renal function, telemetry, as well as histology. So I'll show you some of the data. So these are H&E panels. So this is lewisite exposed animals, and these are POW exposed animals. The control panel is on your left, and the lewisite or POW is on your right. You can see extensive tubular injury. This is 24 hours after exposure. Loss of brush border and injury in these proximal tubules mainly, and the same thing occurs with POW following 24 hours of this exposure. And if you look at renal function, shown here by creatinine in patch 1, sketch 1 mice exposed to lewisite or POW, you can see significantly worse renal injury. And if you look at proteinuria or albuminuria with the albumin creatinine ratio, this was significantly elevated. We looked at urine biomarkers like NGAL as KIM1, and these were elevated as well. And if you looked at the serum or urine, so this is the kidney lysates. If you looked at serum or urine, NGAL was elevated as well in the serum as well as urine and in the kidney lysates. So suggesting that these just cutaneous exposure to either POW or lewisite could inflict acute kidney injury. The question we asked was, well, if there is a systemic response, we know that arsenicals can cause hypotension and shock. So is the AKI due to just the hypotension and decreased renal perfusion? So with the help of Stanley in our O'Brien core, we did telemetry where you insert intraarterial catheters and do blood pressure measurements. And you can see in vehicle treated animals, when you apply the vehicle, there is no significant change in blood pressure. But in the POW exposed animal, you see a transient decrease in blood pressure, which comes back to normal after a few hours. And you can see here we monitored blood pressure for about 8 hours. And with POW treatment, there is a drop in blood pressure, but then it comes back to normal after about 8 hours. So it is possible that this hypotension is exacerbating the acute kidney injury, but it's not the sole reason for the loss of kidney function. We also looked at oxidative stress markers, and shown here is using a cell rocks staining reagent. The red staining in proximal tubules is all positive reactive oxygen species, and the green staining is lotus lectin, which marks the proximal tubule. And you can see significant increase in reactive oxygen species in mice exposed to lewisite as well as POW, and this was all within 24 hours of exposure. We also looked at heme oxygenase 1 as a marker of oxidative stress, and you can see by Western blot as well as immunostaining that levels of HO1 in these tubules were significantly elevated in animals exposed to lewisite, and the same was true for animals exposed to phenyl arsine oxide, this huge increase in HO1 in kidney lysates in these B6 mice exposed to POW or lewisite. We also looked at inflammatory cytokines, and shown here is multiple cytokines using an antibody array. You can see huge increases in IL-6, TNF receptor, thrombopoietin, and these were confirmed by individual dot plots. So significant elevation in a lot of inflammatory cytokines in the kidney following this single exposure to cutaneous either POW or lewisite. We also looked at mechanism, is this causing apoptosis, DNA damage, or autophagy? And using markers for DNA damage, shown here is phosphorylated H2AX, significant increase in these nuclei of lewisite as well as POW exposed animals. And using this phosphorylated checkpoint kinase 1, these were also significantly increased along with this phosphorylated H2AX on Western blotting. Together with that, Becklin ATG7 was also increased, as well as LC3A2 conversion, which is a marker for autophagy in the kidney. And here's again tunnel staining to show that both lewisite exposed animals and POW exposed animals had positive tunnel staining within the kidney, suggesting again DNA damage. And a lot of this DNA damage was autophagy. By electron microscopy, you can see these double contoured intracellular vesicles that are containing lysosomes and these broken up mitochondria with their cristae completely distorted, showing significant cell damage, and markers of apoptosis like BAX and so on are also upregulated in this setting. So it's a combined injury causing both DNA damage, autophagy, as well as apoptosis within the kidney in this setting. So I'll tell you a little bit about this NIH CounterACT program. This was new to me. We were funded by an earlier project with Dr. Madelon, looking at bromine-induced kidney injury on an R21 grant. It's a program that was started by the NIH. It's trans NIH, so multiple NIH institutes fund this program. It stands for Countermeasures Against Chemical Threats, CounterACT. It's essentially to develop countermeasures for bioterrorism and mainly chemical weapons, to get new and improved medical countermeasures to prevent and treat acute as well as long-term conditions that can be caused by exposure to chemical weapons. They support both basic and translational research through R21, U01 or U54 mechanisms. They have clear pipelines required for drug development and regulatory processes, and there's a special congressional appropriation that funds this program. So we were really fortunate that Atar and his team was funded on the first attempt last year with a grant of $18.9 million that funds this program over 5 years. So this is the Arsenical Center at UAB that Atar is the PI. So our goal is the first project is looking at the skin. The second project is looking at the lung. The third project is our project looking at the kidney. There are 4 major cores. There's an administrative core. There is an education core that's run by from the School of Health Professions, Lisa McCormick, and I don't know if Lisa is here, but they have been doing an outstanding job with recording all the presentations, live streaming them, placing them on the websites. We have a big component in terms of educating the community, the emergency response teams, the emergency departments, in case there is a exposure of this chemical. And for example, in a big city, how do emergency responders recognize that, well, this is potentially arsenicals induced, and this is the treatment that should be developed. There's a drug discovery component that is run by Mark Suto at Southern Research, and they've been a phenomenal collaborative team. They have great capabilities for doing high throughput screening of not only drugs and small molecules, but also FDA approved drugs that we could potentially repurpose to be used for this purpose. And then the animal exposure facility is at MRI Global in Kansas City. So these are the main projects I told you about. The first project, the second project, and the third project. There's great overlap. We're all using all the cores. It's only been 9 months or so since we've been funded. It's been really a terrific program. It's a very benchmark driven program. It's not like an R01 that they give you the money and say, come back after 5 years, we'll talk to you later. They have benchmarks that we meet with the program officer every 6 to 9 months, and they go over our milestones. What have you done for milestone A, B, C? You have to check every box possible. They have a CounterACT meeting at the in New York in June that we all have to go and present our work at. So our project is looking at the kidney. So we want to mitigate kidney damage in these animals, at least, that are exposed to arsenicals in the skin. So we want to further characterize the time course, how quickly the acute and the delayed effects occur in mice. We want to find out the mechanism, and one of the mechanisms we are looking at is an epigenetic marker BRD4, as well as a necroptosis marker called RIP kinase 3, and obviously our favorite molecule, hemoxygenase 1, and it seems to be protective in this setting. And then finally, to develop targeted therapeutic intervention to at least protect in this setting of both not only the acute effects, but also the delayed effects of arsenicals induced acute kidney injury in this setting. So that's another talk for another day. We are still, a lot of work is in progress in terms of the therapeutic aspect. We've done a lot of molecular analyses like RNA-seq and so on to see what are the pathways that are being activated. I showed you some of that data in the skin, but we've done similar work in the lung and the kidney, and the signatures are very consistent in the type of the transcriptomic response that is occurring in multiple organ systems following exposure to these arsenicals. So going back to Napoleon, you got that right. How did he die? Any guesses? So he did not die from arsenic poisoning. Okay? So that is very clear. So 200 years later, some Italian scientists who had very high capabilities, highly sensitive capabilities of measuring arsenicals, got hair samples of Napoleon. He used to love to get his hair cut, and they would sample those and keep them. So they had hair of his going back to when he was 10 years old. So they sampled all those hair samples, and all those samples had the same high levels of arsenic when they sampled it when he died. So that when he died, they collected his hair and nails and said, oh my God, his arsenic level is high. So they said, well, he was poisoned at St. Helena when he was captured after the Battle of Waterloo. And that's when they poisoned him, and he died from arsenic poisoning. But these Italian scientists, 200 years later, got access to these hair samples and said, no, that same 200 microgram per gram tissue was there when he was 10 years old, so clearly he didn't die. And they also sampled a lot of his family members. So it just happened that he was either drinking water from areas that were contaminated by arsenic, but he really didn't die of arsenicals. And he had a lot of stomach problems apparently, so they thought he had a peptic ulcer. But at autopsy, he actually had a significant stomach cancer, and from his symptoms that he had lost a lot of weight, he was not eating well, that most likely he died actually from stomach cancer. So the audience got it right. So this was actually new to me, to be honest with you. I thought for a long time he died of arsenic poisoning. So when I read in detail that actually he died from stomach cancer. So, but a lot of famous people have been poisoned with arsenicals and have died. So I'd like to acknowledge a lot of people who've been involved with this work. I'm just a spokesperson for that team. Several people in my lab, and Amy has really taken a leadership role on this collaborative project going back to 3-4 years from now. So really appreciate everything she's done. The Atar lab for really bringing us into this group. several members of his lab, so forgive me for not including all the names. They've been tremendous. Ritesh in particular, the work I showed you, but he's actually the first author on that paper that was published about 6 months ago in AJP Renal. Other members from Lisa's lab, Stanley Feng, and funding sources.